Hello everyone, let's get started. Today we're going to be talking about photosynthesis. Photosynthesis is actually a set of two reactions that are linked together, what we call the light-dependent reactions and the Calvin cycle. In eukaryotic cells, both sets of reactions occur in the organelle called the chloroplast. In the image on the slide, you can see a plant underneath a light microscope, and you can see individual cells outlined, and within each cell you can see multiple chloroplasts. Chloroplasts have a characteristic green color even without staining the plant tissue because of the pigment that's found in chloroplasts called chlorophyll. The two sets of linked reactions that occur during photosynthesis, again, are the light-dependent reactions and the Calvin cycle. During the light-dependent reactions, ATP will be produced, and the electron carrier molecule NADP+ will be reduced to NADPH. Along the way, oxygen will be produced from water. During the Calvin cycle, carbohydrate will be produced using the products of the light-dependent reactions, ATP and NADPH, as well as carbon dioxide gas. These two sets of reactions are linked together by electrons. Electrons will be released when water is converted to oxygen. This is a process that requires ATP. The electrons will be carried by the carrier molecule NADP+ to reduce it to NADPH. And then the NADPH and the ATP produced will be used to convert carbon dioxide to carbohydrate. The image on this slide is what is sometimes referred to as the sunshine diagram. It's a very simple diagram, but it summarizes the key points of photosynthesis. Photosynthesis begins with light energy that is emitted in the rays of the sun. The light-dependent reactions, which are depicted as light-capturing reactions in the drawing, are going to be used to produce ATP and NADPH. Along the way, water will be converted to oxygen. The ATP and the NADPH are going to then be used for the second set of reactions called the Calvin cycle. Those molecules, along with carbon dioxide, will be used by the cell to build carbohydrate. Let's take a couple minutes and look at the structure of chloroplasts. Remember, the chloroplast is one of the double membrane organelles that we discussed earlier in the course. The chloroplast is one of the organelles that is believed to have evolved through endosymbiosis. Chloroplasts are membrane-rich. They have an outer membrane and a convoluted inner membrane, which produces structures that are called thylakoids. Thylakoids are stacked into structures that are called grana. These are the flattened pancake-like structures that we see under the microscope inside of chloroplasts. There is a space or a compartment inside each granum. We don't give it a special name, we use a generic term, and that term is lumen. The space inside the inner membrane, but outside of the thylakoids, also gets a name. We refer to this as the stroma. thylakoid membrane has huge amounts of pigment in it. And again, the most abundant pigment that we find in thylakoid membrane is chlorophyll. It's important to understand exactly what a pigment is because it plays a critical role in the process of harvesting energy from sunlight. A pigment is simply a molecule that can absorb certain wavelengths of visible light. It then either reflects or transmits the other wavelengths. Chlorophyll reflects green light. It makes plants and some algae and some bacteria look green to our eyes because of that reflected light. Chlorophyll is able to absorb visible light in both the red and blue wavelengths. So it's the job of chlorophyll to capture the light from the sun, or the energy in the light from the sun. In order to discuss that process in detail, we have to do a little review of what exactly light is. If you remember from your physics courses, light is a very interesting phenomenon because it behaves as both a wave and a particle, and we refer to the particle in light as a photon. Because it has a wavelength, we consider light as a form of electromagnetic radiation. Remember, all a wavelength is the distance between the top of two waves or the bottom of two waves. So what we would call the crests, the top, or the troughs, the bottom. If you measure that distance between the crest of two waves or the trough of two waves, you can determine the wavelength. On this slide, we can see a scale of electromagnetic radiation according to wavelength. If you start down at the bottom of the image, you can find the radio waves, and these are the least powerful form of electromagnetic radiation on the scale. The longer the wavelength, the less powerful the electromagnetic radiation. So because they're at the bottom of the scale with the longest wavelength, radio waves have the least associated energy with them. And this makes sense. We know that radio waves can travel a long distance, but we also know that they're not capable of penetrating tissue, for example. Above radio waves, we find microwaves with still a relatively long wavelength, but shorter than radio waves. Then above the microwaves, we have infrared radiation, and then we hit the visible light. What they've done in this image is they've taken the visible light range and they've blown it up over here so that we can see all of the colors in the spectrum of visible light and the associated wavelengths. So the red light in the visible spectrum has a wavelength of about 700 nanometers. And the violet light on the other end has a wavelength of about 400 nanometers. Red light, therefore, is less energetic than the violet light. Above visible light comes ultraviolet radiation. We know that ultraviolet radiation is more powerful than visible light, capable of penetrating living tissue, if only to a very shallow depth of about 2 millimeters. Above UV comes the X-rays, which are more powerful, able to penetrate tissue, and then the gamma rays at the very top. The X-rays and the gamma rays are both forms of ionizing radiation. So when we discuss photosynthesis, we're talking about two particular wavelengths of light that are in the visible spectrum. We're talking about red light, which has the longer wavelength of about 700 nanometers, and blue or purple light, which has a wavelength of about 400 nanometers. And it turns out that these are the two types of visible light that drive photosynthesis. Another way to say that is that red and blue photons are most effective at driving photosynthesis. And so we refer to these wavelengths as being the action spectrum. Take a look at the graph on the right side of this slide. On the x-axis, you can see the wavelength of light in nanometers, and it ranges from 400 to 700, again from the violet to blue range up into the red range. On the y-axis, we have the amount of light that's being absorbed, and there are multiple pigments shown on the graph. So the dark green line represents a form of chlorophyll called chlorophyll A. You'll also notice a light green line that's labeled as chlorophyll B. And in fact, there are different forms of chlorophyll in organisms that are capable of doing photosynthesis. You can also see a yellow line on the graph that represents a different type of pigment molecule called a carotenoid. And we'll talk about carotenoids in just a minute. From the graph, you can see that the amount of light that both types of chlorophyll absorb goes way up down in the purple-blue range. Then it comes down to almost nothing through the green and the yellow and the orange until it goes up once again in the red range. Chlorophyll pigment is capable of absorbing light in both of those wavelengths. So inside an organism that can perform photosynthesis, chlorophyll is the main photosynthetic pigment. But there are accessory pigments in those cells as well. The carotenoids are one, and there's another called flavonoids. Accessory pigments actually have two jobs to do in a photosynthetic cell. The first thing they're doing is absorbing light, just like the chlorophyll is doing, but they're going to just pass that energy onto the chlorophyll and not do anything with it directly. The second role of the accessory pigments is equally important. These pigments are considered protective to the cell or the organism. Sunlight contains not only the visible spectrum of light, but also ultraviolet light. And as we all know, exposure to ultraviolet light can be damaging to living tissue. Ultraviolet light is capable of penetrating tissue and knocking electrons out of orbit, creating what are called free radicals. What pigment molecules are able to do is scavenge those free radicals and quench them. That's the language we use to indicate getting rid of those damaging free radical molecules. So, accessory pigments are capable of absorbing light and passing that light on to chlorophyll, and they're also considered protective molecules, almost like a sunscreen for the organism that is doing photosynthesis.